prediction. So let's begin with a recap. 1D, 2D, 3D. We can predict 3D structure essentially by comparative modeling, where we find proteins that are similar. Yes?
Is there no clue to it?
Say again?
Is there no clues to it?
I do not know the answer to your question. It's not open. So then most likely not, because the last lecture was a whiteboard lecture. So the point, the way we do this, is that Dimitri samples the quizzes from the videos of the lecture. And since the whiteboard was not sampled, I suspect that he may not have had a quiz for today. Yes?
Does that also mean that the chalk talk won't be part of the exam?
Not really, and you will see the answer to that question more clearly in today's, because the idea of a short talk is, so typically the way I have my lecture, if I have enough time, is that I have a lot of short talks and I repeat the material in the slides, which is what I'm going to do today. But I feel it sticks better if you sort of do it both ways. Any other question? Again, so for about every other protein, so for half of all the proteins, we can apply comparative modeling. For all the other proteins, we are going to hold what we can do in terms of 3D prediction. We cannot predict 3D structure for the remaining. So again, there are about 150 million protein sequences known. For about half of those, there's one region that I can model the 3D structure of based on comparative modeling, because there's something that I can realize as similar from sequence alignments in a protein of known structure, experimentally known structure. I can map those 150 million then, or 75 million, to the 150,000 of which I have experimental structures roughly. That's comparative modeling. That covers most of the thing that we can do with structure prediction. For the remainder, we can do structure prediction for some proteins, for isolated cases, very short proteins, and some special cases. For all the other 75 million, there is this issue of predicted contacts and doing AlphaFold, but again, that will be toward the end of the lecture, because it is not applicable to all those 75 million. What is applicable to those 75 million as a bunch, as a whole set, is a simplified prediction task, the prediction of 1D structure, secondary structure. And there are two main themes of secondary structure, two main secondary structures, the alpha helix, that I typically use the letter H for, and the beta strand, often used as the letter E for extended. The formation of both is stabilized by hydrogen bond formation. This one in some sense is more local because the hydrogen bond is between I and I plus 4, while here every other residue has a hydrogen bond to another strand, and that strand can be from relatively far away in sequence, forming sheets. Again, any questions about secondary structure as an idea? And so let's get into the secondary structure prediction. So the first actual prediction method, and that was sort of triggered by her question last time, was one that was really saying proline breaks a helix. So that was the rule of a chemist or biochemist. Prolines break helices, and from proline that was sent Gurgi. And again, so remarkable about this, sort of he proposed in Science a secondary structure prediction method in 1957. Later he got a Nobel Prize for something else. But he proposed the secondary structure prediction method before the first structure was actually known. The way we sort of generalize that, we talked about in the chalk talk, is we simply compile a probability matrix where for every, if we had a two-state system, so we write all the amino acids, we write the letters, and we essentially predict the majority probability state. That sort of single residue, highest probability, winner takes it all, simple idea, has been published in a bunch of methods, often referred to as Chow-Fessman, for instance. That was one of the first. Barry Robson here is one of the main authors, and that started what people call the first generation of methods. But simply look at an isolated amino acid and ask what's the propensity for that amino acid to form secondary structure, completely forgetting about the environment. As we already discussed, much better to look at the environment meaning the residues, the amino acids before and after it, and sort of making the secondary structure prediction of a particular L, letter L, at some point depend on what is before or after that L. And that's the idea here. So we are looking at, we are extending this idea by going left and right and compiling conditional probabilities. So compile the probability that this is in an alpha helix based on the reality that these particular residues are next to it. Where do we get the second order structure from? That is the DSSP method. We get it from PDB. We quickly went through that. Now, so this is just the point that I want to make here. The first generation of methods that use simple probability are sort of based on something like fewer than 100 structures. How to assess for performance? We talked about that before. So the simplest way is essentially a match state. A match state simply asks, if I have a second order structure in a 3-state, HEL, how many of my residues are correctly predicted as either of those 3 states? That's typically referred to as a Q3, was introduced in this book as Q3. So this is a 3-state probability, okay? And when we look at the first generation methods, they sort of claimed that they get 63% accuracy. Later it was found that the performance is more in the ballpark of 50 to 55. 50 to 55, is that good? Well, one way of looking at it is more than half full. So think about the 55 is more than 50, so more than the glass is more than half full. Is it good? So one way of, before you answer a question such as this one, the first question you have to ask yourself, what is sort of the simplest thing I can do? The dumbest thing is I throw a coin, right? Sorry, let's not call it dumb. But the least information you can use is by throwing a coin. And that is so what is random, right? Random, if we had those 3 states equally distributed, would be 33. If the states would not be equally distributed, would random go up or down? Say if one of those states is, well, if they are equally distributed, one of them is more than another. Will that increase the random probability? Who is for it? Yeah?
If any of them is more probable, I can always go for that one.
Exactly, that's one way of thinking about it. That's, so you can, again, this is something that you can easily program in a computer for whatever your toy problem is, and you will find out, you can learn it from statistics or you can do it in a computer. It's a simple program that you can do in a few minutes. And you will see that it always goes up. One way you can do that without a computer, just thinking about it, you could just, if you had a majority state, say 30, so we have a 3-state system, 33 is the average, and say one of them had 40%. You could always predict that state, and you already have randomly 40%. So this is no longer quite random. So now we have a, let's call it educated prediction, because you already optimized the Q3. And in fact, so for the way that we have the distribution, the random would actually be 35, and the educated would be almost 50%. But this educated one would be always predicting the state other. And it's not very useful. If you want to do second order structure prediction and you say, well, I have a method that always says there is no second order structure, this is not bringing you very far. It is bringing you to Q3 of sort of almost 50%, which means that this the single residue probability here is possibly not perceived as so impressive, but still, if you had 50% and you could sometimes get the helix right, versus a method that gets 50% and never gets anything, then clearly this is already better. You're not beating random by far, but it's already better, okay? Now, we talked about that in the second generation. What you would do is you would essentially look at segments rather than isolated probabilities, and the math for that has been worked out by Barry Robson again. By the way, he was also behind the first Big Blue that let IBM play chess. The Gore-3 method here, as it is known for the 3 authors, Robson is the last one, Garnier brought the idea of the biology, Oscar Thorpe a lot about the protein, the way proteins behave. Great team. Gore-3, the third method they published, gets to a 3-state accuracy of 55 to 60%. Roughly. But again, looking at conditional probabilities and looking at things that happen from a little bit further away from the sequence. Now, at this point in time, so this is late 80s, early 90s, now this is an interesting problem, and what people do is they essentially apply any kind of algorithm they can think about to this problem. And they always hit it at sort of 60%, 60 to 65%. Why do you believe that is? So let's just say it. The first problem here really is we're not getting to the 100%, no matter what method we use. So this is, mind you, 25 years ago. Or, by the way, but at this point, almost 30 years ago. But what do you believe people argue? Why that would be the case? In fact, so I started my career in this field in the late 80s, and during my PhD I counted over 200 publications in the course of a year. 200 different methods trying to solve this problem. They all sort of got to this limit. Very different algorithms. What could be the reason? Any idea? Well, I give you the task for a thesis, and you're not getting higher. And you try a lot. Why could that be? You could have made a mistake. Okay. But what else could be? What will you try out first? You will try out how in your method some aspects play a role, right? What would you play with? Or what would you look at? Any suspicion? So we go, yeah?
Things like the cutoff values.
What is a cutoff? Yeah, cutoff sounds good, but what do you mean by that?
That you're saying I only need to be 40% positive about this being the right match.
Oh, so you essentially say the threshold that we use for this method that uses probability. That's a good idea. So the way I present this here is a simple greedy approach where we simply take the prediction that is the highest probability. When I say there are 200 different methods, they use all kinds of things to compile the probability model slightly differently, including HMMs. So where we somehow look at the optimization of the probability through the entire model, which means in fact you're not going for the local greedy approach. So this is already part of that. At least some ideas. There are some ideas not planned, and we will talk about some of that, but more generally, yes? So this is one important issue. So you could suspect that it may relate to the fact that you have too small a data set. How could you test that, whether that is true or not? So reality is, no, it's not true. In many applications, in our field at least, we feel the data is too small. Actually, in deep learning now we're getting to the point where suddenly the data sets get so big that 3 months are no longer enough to train the models. So now we really have the feeling we have enough data. We have too much data. But anyway, in a normal application, how would you sort of check out whether you have enough data or whether the limitation of the data is the problem while you're hitting a ceiling on, say, 60%? How would you check that? How could you check that? I give you a data set. You develop a method. That method gets 60%. You suspect that your data is too small. What could you do to sort of, you cannot, so the answer get more data is not a valid solution because you don't have more data. How can you sort of still approach that question? Yeah?
Try to use the same data set, but with different training sets in that data set. And then you see if your results get better.
So you split the training set differently. That's an interesting idea, and we get into the cross-validation story. So let's just make it in the simplest way in which we do it. Say I divide my entire data set, which is represented, the circle represents the entire data set, and I divide it into a training and a testing set. And then I put this testing set away, and use it to see how well my method does. This is what cross-validation does. And let me just bring this idea of cross-validation. The reason for having cross-validation. Some people in this room have heard cross-validation. Can you explain to me why we need cross-validation? I just take somebody else because, yeah.
Yeah, so if the data set isn't big enough, that we have a sufficient...
Oh, you answer the other problem. Are you trying to explain cross-validation?
Yes. But if...
Cross-validation has nothing to do with the size of the data set.
Okay, I think...
You will always have to do cross-validation. Why?
Oh, then
I completely derailed you. Sorry.
Yeah, I think I'm talking about something else then, because I wouldn't split the data set. I wouldn't reuse a training set for testing if I had enough data. So that's where I was going.
Okay. Let me speak out loud to everybody what she said. So she said cross-validation, and we still have to sort of decide why we need cross-validation. But she says cross-validation is something that I split training testing set, and if I really had ample data, I would never use the trained data again for testing. And let's hold that question for a moment, and let me get back to the question, what is cross-validation for?
Cross-validation is to assess how well the model is by giving it different pairs of training and testing data, and to see if...
So how good is the model by giving it different slices of the data for training and for testing? That, therefore I assess how good the model is. Why can I not do that on training alone?
You get overfitting.
You get overfitting. You could overfit your model to exactly one data set. Now, if I looked at the simple amino acid statistics, per residue statistics, when we say overfitting, typically we think about machine learning. We have 20 different parameters. Could I overfit? Yes, we can. And this is exactly one of the issues why cross-validation is something that came into the world of science through machine learning, largely, because it's absolutely vital for machine learning. But it's absolutely vital for statistics too. People just don't take it seriously enough. But it is something that is actually necessary wherever you are. And that goes a little bit on the issue of the size of the data set. Now, let's make a much more generic statement. Whenever we develop a method and we're trying to use one data set that we have today to assess how well the method does, our first assumption has to be that our data that we have is representative for what we will see tomorrow. This assumption is most likely not right. But the data set we have today, we have to see that the data set that we can get is optimal in making that statement. Now, what in this data could make it completely not optimal for that? If it is too small. So if I had, of course, I have the PDB that has, say, 100K, 10 by the 5, and I pick out 2 proteins. That would be clearly wrong. But if I took the entire data set that I have today, is that the right way for being as representative as I can to data set of tomorrow? The answer, of course, when I ask you that way, the answer, of course, is no. Why? Why can I not just take all the data that I have and say, this is the best way, because it's the best thing I can have today, all I have is the best way to hope that I represent tomorrow? What is wrong in that? Yes?
If you assume that, then you can just complete your overfitting the data set.
Then you can complete what?
Overfitting the data set.
No, wait. Wait. I'm not talking. Sorry, I'm going a step back. I'm getting simpler now. I'm not talking about cross-validation anymore. I'm talking about the circle. So now I'm asking, how do I actually bring the circle that represents my data of today? How do I best get that? Do I best get that by taking all the data I have? And my argument is no, that's not the right way. Why?
Well, because you probably find easiest and smallest proteins first, and then the larger and more complicated ones that might look completely different.
So ultimately what she says is you find easier proteins first and then more complicated ones later. that is true, is absolutely true, but it is something that sort of is true for this field. What is not true for this field is that whatever data set you have has some bias. And there's a variety of reasons for this bias. So the most extreme one here, if I now took, so PDB is in the ballpark of 10 by the 5, about 100,000 proteins, 150,000. So factor, give or take a factor of 2, doesn't matter, right? If I now take the following algorithm, I take my protein out of the PDB, and I ask, is there any protein in there? So for the second one I add to my list, I ask, is it more than 99% sequence identical to the one I already have, to any of the ones that I have already? Or more than, let's make it more extreme, 100%. So do I have exactly that protein in my list already? If I do, then I don't take it. Any idea how much this 100,000 would go down? If I just kicked out 100% identical, how much would be left? Any idea?
I have a question. If they are 100% identical, why are they in the database?
So you're going two steps at the same time. You're saying many, much more than I expect, is why you asked the question, and that is correct. So 50% is gone. So your next question, why is 50% gone? And the answer simply is because people want to see whether you get the same structure with or without binding a substrate. So if you wanted to develop a drug, then you want to see how that drug actually is changing that protein. So you're going to have two structures, one with a molecule bound to it, and one without a molecule bound to it. And there is a lot of these studies, and that explains why about half are identical to something else. Now this half is not, this is not done for every single protein. It's just some proteins, very much along the lines of what you said here, some proteins are easier to study, and for those studies are more frequent. They simply study the effect of binding upon protein structure. And that is done, so there are some proteins that are more than 1,000 copies of that. So taking this entire set is absolutely wrong, because you do not have a good representation of tomorrow. You simply sample, how do people do experiments today? How do people, or what, anyway, so the machine would learn completely the wrong statistics. So at least you have to do, this is not all. There must be a redundancy reduction. Okay, that's the first important step. That's long before cross-validation. But it has to do with cross-validation, because the essential assumption in cross-validation is that my circle represents tomorrow. Otherwise, there is no point in split, in any split. And that's why it's sort of a step before I even talk about cross-validation. Okay, now we get to cross-validation. We get to this issue, and that is, in fact, we get to the next point. Now I believe that my circle here is the best I can do to represent the data of tomorrow. And now we're getting to your point a little bit. No matter how large that is, if I believe that this one here represents tomorrow, and I have a particular cut, I'm not entirely sure that this cut here is completely representative of tomorrow. I know the whole thing is as good as I can. So that is why I'm going to use one half for training, one half for testing, one half for training, and then I turn it around, no matter how large the data set is. Because only then can I trust that I really sort of sampled the diversity. And this is why, in fact, the split is independent of the size. Now back to the suggestion, and I'm sorry that I go slow today. The suggestion was, maybe the issue with do I have too little data, I would address it by having a different split. Now, in this different split, say you get the same result. What does it tell you? Does it tell you that the data set was too small or too big? Oh, let's say the results are the same between, I'm not responsible for that. Let's say between split 1 and split 2, you get the same result. What do I learn about this, whether the size of my data is the reason why I only get to 60%? Do I learn anything? Oh, be careful. The size of the data set is common knowledge that if I put more data, then I learn more. That only depends on how much information I can extract from the data. And maybe, for instance, there's a variety of, if you think about machine learning, there are all kinds of other things that could go wrong. And for instance, my input space, my feature space is so uninformative about solving the problem that actually I cannot extract anything than sort of the simplest linear correlation. And then the size doesn't matter, because possibly I get the linear correlation when this whole thing says 50,000, I would get the correlation out of 5. We have many of these examples. And actually, very often, these complicated machine learning packages do not tell you whether you're actually using the entire diversity of the data or need to. But anyway, so this doesn't help you. How could you? Back to the question. You suspect that the size of the data is the reason, or the smallness of the data is the reason why we're not getting over 60%. How could you check?
Make it smaller and see if it's getting worse.
Exactly. So you just take, for training this out, or more extremely, you take 1/10 out for training. And you see if you train with 1/10, do you get the same performance? If you do get the same performance, the size of the data is absolutely not the limiting step. That much is clear. Now, in those days when the 60 were reached, that was not something we could do because the size of the data, that was 10 proteins, that was too small. And when you sort of took out a third of the data, the performance again looked pretty much like 60. So it looked at this as if the size, sort of somehow, just a sec, suggesting that the size of the data would not be the problem. It turned out this was not a right statement, but so this suggested that this is not quite an answer.
So was there a split back then or not? I mean, they had to do some split because otherwise you cannot prove that.
Yes, we had a split, and in this split, what we could try is to take out a third of the training set. Otherwise, the training set became too small. And the conclusion from that was there was no change. Yes?
Okay, and how was the performance on the training set? Because the statement, like 65%.
And now we're getting into something else. So could I train and overtrain that? And that is a very, very good question, and it's something in particular for machine learning that you always have to look at. Can I actually train it? Whether it's overtraining or not, can I at least, with my way of doing the input space, can I even overtrain the system, right? And the answer to that question was, in those days for the machine learning devices, you could overtrain not to 100%, but you could overtrain to values much higher than 60%. With the statistics, you did not actually get much higher. You still sort of were bound at 65%. What else could be the reason? Yeah?
This sounds a bit like there's just randomness in the training set.
There is randomness. Absolutely right. Where could that come from? So the randomness, of course, is a randomness somehow of the input with respect to the formation of secondary structure. Where could that come from? It's totally right. It sounds like you could say randomness, you could say there's something fishy going on. Whichever way you formulate it, what do you believe could be the biological or biochemical explanation for that? Mutations. Yes, you and your neighbor have one variant change in every single protein that you carry. But in this particular case, we're looking at the same protein, so there's no mutation going on. In this particular case, that is not the answer. What could it be? So you're looking at individual residues, or you're looking at segments. Going from individual residues 50 to 55, you go to 55 to 60 if you look at segments. Segments typically are sort of 13 consecutive residues. What could go wrong? So you go up 5 percentage points by going from single residue to sort of residue embedded in an environment of 13. Yes?
Maybe if you just go over a random number, 15, 20,
Right, so that's the first issue. Make this segment larger. And when you make it larger, what you see is that you're not improving. You're actually sort of hitting, for some methods the optimal point is 11, for some other methods the optimal point is 17, so that actually depends on the algorithm, what the best point is, but you're not getting much higher than around 13, 14, 15, or some number like that. And that, in fact, is partially an issue, again, clearly of numbers, so of the size of the data set. So the bigger the data set, these numbers get higher. But what is going on in terms of the biology of the problem? Anybody has sort of an idea? Why do we actually want long segments? Why could it help to have longer segments? Yeah?
Because the protein falls into a global optimum.
Exactly, the whole thing falls as a global thing, and secondary structure itself is not fully globally determined. I said that the secondary structure formation of a helix has the hydrogen bond formation between I and I+4. So in a window of 30 consecutive, you see the hydrogen former. In a beta strand, you do not see the hydrogen bond former. What do you therefore expect in terms of prediction? So alpha helices are more local than beta strands. What you use here is a more local signal. What do you expect? Yes, it's the opposite. Is that why you're smiling?
I'd say you're hitting local optima with your algorithms.
Yes, you hit the local optimum. That's in fact in an energy landscape. Surprisingly, so when I trained in the first neural networks on this, I was very surprised to see that I'm not stuck in local optima. So that was not, in fact, this is not the way I talked about. In this sense, I did not use the word local, but I actually suspected exactly the same thing. That's why I checked very carefully when I first applied neural networks to that. I couldn't understand why I was not stuck in local minima, but anyway, I wasn't. So that is not the issue. There is something about local. So, okay, now. One way in which we can measure the performance of secondary structure prediction on a per residue level is this Q3, the overall match of one of three states. Now, we talked about it last week already on the whiteboard talk, that what the problem of such a method is, it ignores the fact that some methods may get a helix right, and that may be very important whether or not you get the helix right, or whether at least you say this particular protein has 2 helices and 2 beta strands or something like that. It may not be that important if you have a beta strand E and another one here. It may be good enough if your prediction says a single E, although you know there cannot be E's. But if a single E is right, to somehow see there is a core of a beta strand, or you actually have 2 in the protein. So for using that may be more important than a method that gets all or almost all the non-other states right. How would we measure that in terms of performance? Q3, so say 50% of all the residues are in fact in the state other. So Q3, by doing always O state other, would be 50%. And that is the issue that we talked about earlier. But it is therefore not, important. Q3 is a simple number, an important number. But it's not really telling whether I get helices or strands. How can I change that? Very simple. We talked about it last week, I thought. The confusion matrix, yeah? So we talked about the segment overlap, and that's true, but what I'm hunting for here is a much simpler one, QH and QE. So essentially you're asking how many of the helix residues are correctly predicted, and how many of the beta strand residues are correctly predicted. And so what you would expect, since helix is more local than strand, you would expect that the QH is much higher than the QE. Okay? And that, in fact, is the case. The QE is below 40%. In fact, for some methods, the degree to which they predict beta strands is less than random. So you may have heard that if you have a prediction method that is less than random, you actually have a prediction that is better than random. Why? You just switch it. You just invert it. Can you do that here? So if your beta strand prediction is less than random, can you invert the method? Yes, you can, and you would get a beta strand that is better than random, but you will most likely get a prediction method. You will get a prediction method that is worse. That's the problem. By switching every state exactly around, if your method is better than random and only the QE is below random, then you will in fact create a method that is less accurate. That does a better QE, so this sort of cure doesn't work. Okay, you cannot cure one state in a 3-state prediction. I mean, you can do that, but you're actually hurting yourself in terms of the Q3, or in terms of the overall accuracy. So this really is a problem, and the problem really sounds as if clearly there's an issue of local versus non-local information, and that maybe people argued that maybe the reason why we're not getting to 100%, because beta strand or alpha helix, there is some of the long-range interactions that you need to know when the protein folds that determine how eventually the entire secondary structure looks, and that is what we are not seeing whenever we put a local window. When we make the windows longer, we get too much noise. We get more information in there, but we add too much noise, so the methods, whatever they are, cannot sort of see signal to noise. The signal-to-noise ratio decreases, and that is not good. That's why making it longer doesn't work. But somehow that information is needed. Okay? And here's another issue. Another issue of these early methods was that predictions tended to be too short. So I show you a typical prediction of the first generation or second generation, I cannot remember which of the two that is here, where you see this is the observation, and I told you that E's are typically too long. This is sort of, let's just assume that this is an invention. It's not quite true, but it's too complicated to explain why that is the way. And this, by the way, is red here. This should be H. This is HHH, should be red. So again, H is red, blue is E, and you see that somehow the E is right here, somehow an E is right, so you get some of these segments right here. But since the ones that you have are all very short, you do not know whether that means here a short helix that really is a helix predictor that is right, or short helix is that something you should remove from the prediction in order to beautify it or to make it better. So two short segments are not really meaningful or not really helpful. And that gets to this concept. That is the point where people began to use, in fact, machine learning to try to address all 3 problems. Let's begin with the simplest idea of the neural network. So the neural network in this artificial neural network idea is simply something where you have an input layer and an output layer. You connect the units, and then the simple arithmetic is you multiply the input to this unit here with a connection. You do the same thing here. This green unit, the output unit, upon entry sums. So J1 times the input 1 plus J2 plus the input 2, right? That's the input value, bless you. And then there is a nonlinear trigger function. So essentially what you do here is you have the vector spanned from the input and the vector spanned by these connections here, and you do a projection of one vector onto the other one. Now in the next step here, you have the sum of all these products that come in, and you apply a trigger function. You simply say if this sum is very large, you switch it to 1. If it is very low, you switch it to minus, highly negative, you switch it to 0, and then some function in between. That's a sigmoid function, tangens hyperbolicus, or you could have a linear function in between. Typically people use something that can be easily used to compute, and the E function here is typically used. This one is good enough. Now, essentially what this one then does, if you want, it sort of makes a decision whether or not this product of these 2 vectors is smaller or larger than some number. And what you can do is you can classify problems with it. For instance, you can sort of classify these 2 types of points, the open circles and the dark circles, and you do that by sort of reducing the error. So you have some connections that you start with, and you simply say, well, this should be switched to 1 and this should be switched to 0, that means the actual output that you get from this device gives you an error. Or gives you, you know what you get out, you know what you want out, and the difference of these two, you square it because you want to make it sign independent, error cannot be negative, and then you simply sum over all the errors, and then you apply an algorithm that simply goes down in the scale of error. So you change the connection such that the error decreases. It's simply a gradient descent. Okay, that's the simple idea of a neural network, and as simple as that is, so I show in detail the training, and since it's a gradient descent, there is no clear solution. You find different solutions. In this particular case here, I show 4 different solutions. These solutions introduce essentially a line in the space, and each of these 4 classifies, in this particular example chosen, completely correctly classifies the point. And they are, in that sense, cannot be distinguished. Now, how could you do that with this problem here? Show me the line that classifies the open and dark circles in this one. Yeah?
Either you have 2 lines, which need to be classified both sides, or you have
Yes. I'm sorry, I thought the first image was the 2 lines. But I completely recognized that you were going the right direction. So it's just that my image is the wrong way around. I should have first shown the 2 lines. So you can either do it with 2 lines here, or with a, and I'm sorry for the animation, I have to repeat the animation. Okay, you saw it. So I can, since I didn't say straight line, you can have a line like this. But the line like that, in fact, what the system here really does, it introduces straight lines. And you cannot introduce straight lines with what I showed you so far. And for that, you need another layer. And that layer is called a hidden layer, simply because this is input, that's real world, that's output, that's real world, and the red one, the in between here, is hidden, it's just computation. The way the computations work, totally the same as before. You simply add up the input, you apply the trigger function, take the input, multiply it with the connection, sum it up, sum it up means from all that's come in here, apply the trigger function, use that as input here, multiply it with that connection, sum over the lines that come in here, apply the same trigger function, and you have the output. Really very straightforward. And then there's sort of some issues still not here. The error function could be this one, could be something else. Now, what could happen with what I showed you so far is you could get stuck in local minima. So you could stuck in this one. You prevent from getting stuck by allowing for every single move step a jump up. And that is essentially, so this is the term that I show here, the derivative of the error with respect to the connections here. Minus means you go down, so the change in the next time step is proportional to this derivative, downhill that means here, plus some uphill move. And this uphill move is simply what you take from the step before, the change that you did on the step before. Totally trivial. Now, there is one sort of little thing here. In reality, you have to compute this error function for individual samples. So for most problems, so the XO problem that I showed you before, open and dark circles, there you can easily do that for batches, but in reality for real problems, for millions of data points or 100,000s of data points, you essentially apply this function sample by sample. So you present the samples one at a time, and you then, for every single sample, so time somehow is given by presenting one sample and changing your error, or changing your connections accordingly. And that gets us to this point. When I show training time here, and I show here the performance that you reach, I have 2 lines. People already talked about that here, overfitting. The green one typically is the training set, and the blue one here is what we consider to be the testing set. So in this particular case, where would the overtraining be? Or would there be an overtraining? Yeah, already, you saw it in your... The funny thing is, before I actually put the hands, your face was already pointing to it.
Like where the train arrow drops, but where the train arrow increases the train. Jesus. So where the test accuracy drops, how the train accuracy still increases.
Yes. So this is sort of the point here, where the testing is falling and where the training is going up. Was that another point? Yes.
So the y-axis we have the error.
Training time. So training time could be. On the y-axis you have the accuracy. Yeah. So error would be 1 minus that. Yes. Otherwise, right, that you sort of have a reinterpretation of my plots. So I switched between before I showed the error on the y-axis, and now I show the performance. You're right.
And the blue one is the test set.
The blue one in this particular case would be the test set. Thank you for asking. Because the set for which you overtrain and can overtrain, it was asked before, can I actually overtrain my set? That is an important question you have to ask. Why could it be that you may not be able to overtrain? Exactly, if you don't have the right input features. So technically the way he formulated it is, if your model does not have the right capacity. Essentially what it means is you have no good feeling of how you can, from the input, learn the output, because you did not use the right input features. That could be a reason. Or again, the data could be too small or too diverse, or there are other reasons. Okay, but back to that issue, then the idea is that you can train, in this particular case, to almost complete accuracy, but in doing so you see that if you take an independent set at some point, you get to a maximum number, meaning after that all you do is overtrain for the green set. Now, what I have to tell you, this is a toy problem. This is the way we present reality. This is the way we have never in our group seen reality. I will show you a bunch of real problems. It's always much more tricky to see what overtraining is. It's never that simple, but this is the way we typically see it. And now get back to the experimental data set. And that brings us to a new issue here. The new issue is the one we so far talked about, the training and testing set. And we need to use the test set. What do we actually use the test set for? The train set we use to optimize the model. What do you use the test set for? There's no trick in the question. Validation. Validation means if I had an example where I have method 1 reach a Q3 of 60% and another one, method 2, where I have a Q3 of say 65%. And say this method 1 here would have used 100 hidden units, and this would have 50 hidden units. My first question is, which set do I take that number from? You said validation of the method, that's too abstract. Here I have a choice problem. Do I take the training? Is the 60 from the training set, or is the 60 from the test set? Or 60 or 65? Which set do I use for this? Yeah? Always the test set, because we're trying to have the set that is independent. Okay, now I go further. I see that 100 hidden units is different, performs differently, and say we will get to the standard error issue, standard error is plus minus 1%. So let's assume that is absolutely statistically significant, the error difference here, and that I can clearly say M2 is better. Then I conclude that M2 I'm going to take for the future 50 hidden units, and my performance is 65%. Why is that wrong? So this is what everybody does. This is what you find in 95% of the manuscripts talking about machine learning. It is absolutely wrong. Why? And again, the answer is not simple, otherwise 95% of the people would not do it. Yes?
Yeah, because I learned the new model on the data I want to test the model.
So ultimately, the important statement, so he says, let me just repeat for everybody, I learn the new model on the data that I use to test, to validate, and I should not do that. Let me sort of absolutely correct, just a moment, let me just rephrase it. The idea of the test set is the holdout set that I do not use for anything else other than establishing the final estimate for performance. Now, I did two things here with that number. Number one, I chose number of hidden units. It's a so-called hyperparameter. So that's one thing. And without changing the set around, I did another thing. I estimated performance. And that I cannot do. Ultimately, it's absolutely what you said. It's just reformulate. Test set you can use only for one purpose. Final number. No comparison, no choice of hyperparameters. How could I get out of that problem? Because I have to choose hyperparameters, yes?
So usually you split your data in 3 sets.
Exactly. I'd like to call, again, in Wikipedia and everywhere else you will find different names. Now, different communities use different names, and that's why I sort of brought in a new name. So everybody sort of understands the same under training set. But some people use the word validation set for this, and some use the word test set for that. And so by calling this one here cross-training, this is very simple, it's very clear what I mean. It has to do with training. It optimizes not the model, but it optimizes hyperparameters. It's something that is used by calling it cross-training set. You sort of have the memory of it being used to do something to the model, whether it's hyperparameter or choosing the parameters, while this one really is setting the test set. If you all you need to do is understand what the 3 sets are for. If you get the names of whatever you want, it's fine. It's just very misleading that some people use the word test set for one and validation for another, and some groups turn it around. Anyway, so you need 3 sets. Yes?
Earlier for the secondary structure, you said you can't have a holdout set for the test set. So
I did not say you cannot have a holdout set for the test set. All I said is you have to, and in fact this is an issue that is not on my next slide, but maybe on one of my next slides, but no, will come later. So when you give the final number in your manuscript, in your thesis, in your paper, that number you create by doing this. In this particular case, you would have to do it 4 times. Such that the test, that every single protein in your data set has been used for testing exactly once.
okay, because my question would be, so for other statistical data on populations, maybe you can have a test set where you have a distribution that you know you're looking at. So for protein, is there anything like it, or?
So are there, so actually, so you say for populations you could have a holdout set that really you know is completely different. And it's the same thing here. So if I had a new data set, for instance, of something that sort of I know represents the reality of the future as well as what I have today, or I have 2 ways of looking at that, then yes, you would in fact look at these 2 different data sets. But I would still argue it is very helpful if you use them both as the final test set once, just to see whether they really behave the same if you sort of change the entire machinery underneath. So I do believe that this cross-validation idea is something that would in some sense also help population analysis a little bit.
Yeah.
Let's turn that around.
It's definitely used. It's just a question of whether I would say, have enough data to just have holdout sets or not.
So again, I'm not entirely sure whether you ever have enough data. And ultimately the issue always has to do with what I talked about when I talked about the cast. So I hide something under the table, but I'm doing my work and it's hidden. I know it's hidden, but oh yeah. It's so easy to do this. And then you have the feeling that you do have enough data. Even in population, you're not creating new data every day, right? It's not that something completely of the same size comes along tomorrow while you're doing so. You cannot set up your method and then look, oh, I have another set that has the equal size, and I can read it. I had never seen that before, completely new to me, and now I can assess my method. There's almost no feel like that. If you are in this situation, that is great. But in most fields it's not like that, even if the fields believe they have enough data. Anyway, let's just stay there. So there's some idea of rotation, there's some idea of splitting. Clearly, this here now, let's make it clear. For the choice, this has to be from the cross-training set. So this would have to represent cross-training, and then you can say, okay, M2 is best, and 50 hidden units are better than 100. Okay, now back to this particular example that I put up here. How can 50 hidden units be better than 100? 100 is more. That's exactly the issue of overfitting again. That typically would be the reason for this observation. So back again, in this particular case here, testing is this one, that is training, and cross-training in this particular case has not been used. Okay, now, and that, so what I do show here in this particular example, I show it and I go over it. So typically in a toy problem you see this clear difference, right? In a real problem you see something like this. In fact, that is the particular method that for which a neural network for the first time predicts secondary structure. And so where's overtraining? You could argue, well, the first time that you're going down, overtraining is here, right? You could argue overtraining is the maximal value that you reach, which is this one here. You could possibly even argue, well, this one here increases, at that point here, this one increases more than that. That's sort of the first indication of overtraining. But you see, this is, in fact, there are mathematical ways, take the optimal and then you have a solution. But it's not quite clear that this, in fact, is the best point. So you would actually want to do this here using the cross-training set. You have to do that doing the cross-training set. Sorry, that is another point that is wrong in my coloring here. You cannot pick the point of stopping using the test set that you will use for the assessment. The early stopping, so deciding, well, say this is the best point here. If you decided that, then this would have to be the light green set. I made a mistake in the way I presented that. Is that statement clear to everybody? It's a very essential point. And I gobbled it up in my talk. Is there anybody who has an issue with that? Okay, let's get back to the secondary structure prediction method. I said that methods did not get to 100%. They did strands sort of randomly, and they had short segments. Now I'm going to use a neural network to predict secondary structure. And this is the way it does it. So I take out 13 adjacent residues from a protein. Each of these blurbs, these yellow blurbs here, represents 20 amino acids. And this is a sparse vector. Hot encoding today, in today's language. Sparse coding in those days. So essentially the vector is such that if it is the amino acid P, then the P is on, all others are 0. Are there alternatives to hot encoding or sparse encoding? What else could you do? So I have every one of these 13 amino acids is coded for by 20 input units. Could you do it differently? I'm sorry. There's no, the way I look at you, there's no difference for you to. From my angle, you were both exactly in the same. I have seen him first there, but yeah, you spoke first. So.
Discrete encoding, just give one number to every message.
So essentially, you could put discrete encoding. So you put a real number, or 1 to 20 integer, whatever you want. Essentially, every single amino acid is represented by one input unit with different numbers. Right, so you have, instead of 13 times 20, you have 13 input units. What else could you do? Yeah?
Some sort of feature vector.
Yes, you could put in something such as, some amino acids are large, some are small, some are positively charged, some are negatively charged, and you could reflect upon that by having, for instance, a vector that has one component for charge, one for hydrophobicity, and blah, 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 blah. And then you do either a 1 or 0, or you do, again, something sort of more a real number. What else could you do? Well, in principle that is it, but you could sort of pre-put some probability into this already, where the input essentially is something where you multiply with the probability of this to form a helix. So you sort of have a mixed model in some sense. Okay, now how do you find out which one is best? Essentially you try them all. And when you try them all, this has to be again the cross-training set. In order to say that, for instance, the sparse encoding versus using a single number, one is better than the other, you have to test on the cross-training set. It turns out sparse encoding has been so far the best way of doing it. Yes?
Did you just put Q3 there, or is that what people use to compare it?
I put Q3 there because this is what is simplest to say. At the end of the day, you would also say, okay, this one here looks better at Q3, but is it exactly one that has the helixes and the strand also correctly predicted, or is it always predicting the other state or something like that? So you would have to look at other features. So I simplify a lot here, and that in fact brings us to the challenge of the game. It often is the situation that you have an example where method 1, so you look at QH, and you have sort of the inverse of the story. So your QH would be 63 here and 61, say. So where that method here is clearly better on this one overall, and that one is better on helices. And then you're sort of left with the question, which one do you now take? And this is a tricky decision that in development you often have to do. What could you do in order to sort of escape making a decision on that note? I'm constantly looking at the time. So being lazy, saying, well, okay, I don't know. I mean, I could ask my user what they like, but the user is not talking to me before I have my method up. So I have to make a decision before I put the method up. So I put them both up. That makes it complicated if 2 is all right. But if you suddenly, because of a variety of different problems, you also want to look at QE, you want to look at segments. So maybe you're going to have 5, 6, 7 methods up, and then users will no longer be happy with you. What could you do to avoid all of that? Yes?
Just like average.
You average. Now, say I have one method and I have an average over n methods. And I'm claiming that the average over n methods, okay, there may be an issue with CPU. Producing one result may be so long that I cannot actually afford to often do n. But say CPU were not an issue. I claim that an average over n is always better, almost always better. Why? Yeah?
Generalized pattern frequency data.
So that's an interesting point. So, but why actually? The statement here is it generalizes better. Why? In principle, our ability to generalize is measured, say we can cut out these numbers here and look only at Q3, let's simplify the issue, then my generalizability is captured exactly by that number. Right? So I have measured that one generalizes better than the other. Yes?
So you've already initialized the weights of your network randomly, for example, so they might have the same actors, but they have very different weights.
So let's try something like this. I have another method that also reaches 65, also 15 units, but it has, for instance, just a different randomization of the initial weights, and you get sort of to the same performance. What you actually observe is that the performance for particular protein is not the same. So for every single protein, it's actually a slightly different prediction. Overall gets the same 65. Which one do you use? The average over both. And again, my question is, why is the average over the two, which in this particular case will be 65, still a better performer? You wanted to say something. I cut you off. No?
Maybe the first method captures some features, the other method some other features, and then you want to capture both.
Yeah, that's exactly why I suspect this. So that is the reason for saying is generalize is better, because one method captures some features, the other method captures some other features, and both together capture all features. That is sort of, let's call it an emotional way of arguing. It is true, but overall, in principle, the generalizability is measured by this. And the generalizability strictly doesn't change. So overall the point, or put it differently, one way of saying that is, as long as these methods that you have are not correlated, meaning they're not getting exactly the same kind of mistake, so they're not always doing the same thing, and as long as some part of your error is random. So the error will always, physicists talk about an error being sort of white noise and systematic. So where there is some function here, the plus is the total simplification of the issue, but part of your error really is a white noise component. So white noise means essentially this is completely random. And this could be, where could that come from? Your white noise in this issue here. Yeah?
Errors in the data.
It could be simply incorrect measurement. That could be part of white noise. It could be, so if you put the protein into a slightly different environment, by putting DNA next to it, by putting it into a slightly different cell type, it may change it a little bit. That is for you white noise because it's not part of your input data at all. The same sequence. So you cannot see that as noise, right? For you, that appears as white noise. Systematic noise would be if your input vector is not long enough to capture all the relevant data. That's systematic, because it's built into the way you construct your model. That's a systematic error. And as long as there is some white noise, if you have a different solution, it hits the white noise. So the generalization, the better generalization that the two of you talk about, is always addressing the white noise. It's never addressing the systematic error, yes?
But does this also hold true if on two models? Because if you average two, you don't know which one.
No. For two, there is not much of an average. I completely admit that this n here has to be larger. But remarkably, actually, averages over 3 tend to improve already. But then, in fact, it turns out the larger n, the more the improvement. You may sort of saturate, but even a few years ago, people published that you can really even do it to 10,000 different models. And that still gives us a slight improvement. And simply better, as long as you have enough CPU, better is better. Okay, I lost my thread. We are at this question, alternatives to sparse coding. All the alternatives we have talked about so far didn't work. And ultimately, why is that so? Because it seems that by using sparse coding, you put the fused assumptions into your system. And by putting the fused assumptions into your model, you give the model the optimal way to learn the problem. And that often, in fact, is an issue to do. However, in this particular case, sparse learning of my neural network still only gets to 62%. 62% is essentially what we got before. So the machine learning did not improve very well on the first issue. It did not overall improve. So just plugging in machine learning is not enough. And at that point, I looked at the way that the system trained. And I observed this one. This is time. Now here, every step is sort of presenting all the samples. And this is performance. That's the Q3. And I'm showing separately the 3 different states. other in green, alpha helix in red, and beta strand in blue. What do you observe? Yeah?
Again, maybe we have a problem distance.
I'm not sure that I understand what you said.
The strand always has less performance than the helix.
Strand is always, oh yes, I did not, I'm sorry on the slide before, I may have made a mistake. We are overall getting a Q3 of 62, and the next question then is strand, so first of all, it doesn't, remember there were 3 problems. Strand is almost random, we're not reaching 100%, and segments are too short. First issue, we're not reaching 100%, we're not improving by machine learning over that. Next observation is, we're not doing better on strand either. Strand is predicted worse. And in fact, that's exactly what you see here. Strand is predicted worse. On the over time, blue is always below the others. Yes?
The sum of the points is basically always the same, so they just complement each other, but there is no gain.
Somehow the system seems to learn something, but whenever you improve one, it is at the cost of the other. This sort of seems, I have not really looked at the number, but it looks a little bit like that. Yes, but something else. So when you write your thesis and you produce your figures, in results you talk about what you see. What do you see? And you have all said things that are important. Blue is below. There seem to be some sort of sum. What else do you see? Look at your figure. I still want to sort of finish that. Give me a few more minutes. Yes? So you're already doing very well after one epoch. And by the way, this is an example where, so you reach something very high here and you're not really getting over that much over that point ever, or not getting over that point at all really. But that's not true for everything. And that's the crucial observation. It's not true for blue. So it sort of seems to be already here at this first point, but not for blue. Blue goes up tremendously. It goes up to something clearly below random, where the other part is already learned, to something that is clearly at least better than random, right? And even more. So it looks as if in the first 1/10 of its training time, the network learned Helix and other, and the remainder of the training time it spends on strand. Why could that be the case? It could be more complicated, yes. But it could be the case because of something much simpler. It could be the case because when I look, 100% of that circle now is presentation in the database. Most of my samples are white other. About 30-something% are Helix, red here, and about 20-something% are blue, beta strand. What you actually sort of have at the end of the day in terms of performance is this. Now, so here's, this is prediction performance and this is database presence. So here I ask, of all the correctly predicted residues, how many were helices, beta strands, and other? And here I ask, of all the residues in my original observation data set, how many were helix, beta strand, and other? These two things look very similar, right? What we want is something like looks like that. How could we get that in terms of prediction? Well, instead of picking randomly my samples, I could take a helix, a strand, and at random another residue at every single time point, and then change my connections, and then take, again, all three. Which means I will oversample strand, and I will oversample helix in training. I will have a balanced training. So this is the unbalanced training, and this is what happens in the balanced training. The red disappears behind there because it's exactly the same. Now that means that you get a performance which looks like this. The overall performance is lower because you actually do worse on the majority class, other, but you get a performance that is absolutely identical on all three secondary structure states. Now, this is an important point. We said, no, all the experts in the field said that beta strand is predicted less accurately because it's less local. Do you believe that statement? Looking in this slide? No, it's wrong. So the machine learning device, by understanding how the machine learning works, by understanding what your device tells you, in this particular case you use the machine learning not to learn something, but to learn something about the problem. And what you learned about this problem here is that in fact, although it makes a lot of sense that beta strand formation is less local than alpha helix. It's just the truth, it's a fact. But that did not lead to a less accurate prediction. You simply present them in equal numbers and you get exactly the same performance for all three. The preference for beta strand formation when you put at windows of say 15 consecutive residues is exactly the same as for alpha helices, as shown here. So essentially this shows you that with machine learning sometimes you can learn something about problems and sort of correct textbooks.